Hello. Am I audible? Hello. Hello. Good morning.
Morning, ma'am.
Yes, good morning, ma'am.
Hello. I hope I'm audible to everyone.
Yes, you are audible and visible. Please continue.
Good morning, everyone. We are transitioning from discovering medicine by chance to designing cures by code. With the divine blessings of His Holiness Swamiji, I extend a very warm and cordial welcome to each one of you present with us today. I, J. Tejaswini Reddy, a research scholar from the Department of Pharmaceutical Chemistry, and it is my pleasure to be your host for today's session. It is my privilege to welcome you all to the final day of our 5-day online faculty development program on artificial intelligence in transforming drug discovery research. which is being conducted from 27th July 2026 to 31st July 2026 and organized by Center for Advanced Molecular Design and Simulation, CAMDAS, Department of Pharmaceutical Chemistry, JSS College of Pharmacy, OT. For the past 4 days, we have immersed ourselves in a fascinating world, dissecting intricate AI models and exploring the frontier of rapid molecular synthesis. Today, we gather for the grand finale of this inspiring learning experience. The upcoming session is on AI in drug discovery, generative AI in molecular design and synthesis by Dr. Rajneesh Kumar. This session promises to tie the advanced concepts together and provide you with actionable insights for your research and teaching careers. Advancing to the core of today's program, now I request Dr. Gomati Swaminathan, Assistant Professor, Department of Pharmaceutical Chemistry, JSS College of Pharmacy, UT, to kindly introduce our esteemed speaker, Dr. Rajnish Kumar, and take the session forward. Please, ma'am.
Thank you, Ms. Tejaswini Reddy. A very good morning to one and all. It's my immense pleasure to introduce today's resource person, Dr. Rajneesh Kumar, an associate professor in the Department of Pharmaceutical Engineering and Technology at the Indian Institute of Technology, BHU, Varanasi. He is a recognized expert in AI-enabled drug discovery with advanced training and notable achievements in neurodegenerative diseases therapeutics, particularly for Alzheimer's and Parkinsonism disease. He completed his postdoctoral training at the prestigious Karolinska Institute, focusing on the preclinical discovery of lead candidates for neurodegenerative disorders. Further enriching his expertise, he conducted research at the Institute of Protein Research, Osaka University, where he worked on predictive modeling of ADMET parameters. Dr. Kumar's research integrates AI-enabled predictive modeling, visual screening, and high-performance computing to expedite the discovery and optimization of therapeutic leads with a strong emphasis on developing a small molecule that inhibit protein aggregation. This is the key factor in neurodegenerative pathology. His innovative approach extends to diagnostics as well. He is advancing PET and SPECT trace development for choline acetyl transferase imaging, aimed at detecting early biomarkers of Alzheimer's disease. With an extensive publication record and funding from both the national and international agencies, Dr. Kumar's contributions have been recognized with numerous accolades, including the Young Scientist Award 2023 from Bioinformatics and Drug Discovery Society at CSB DD 2024. SRM Institute of Science and Technology, Tamil Nadu, and DB Dr. Young Investigator Award presented at the 15th International Symposium on Cholinergic Mechanisms in Marseille, France. Dr. Kumar's advisory experience spans academic and industry platforms where he consults on AI application in drug discovery. As an advisor, he brings a deep interdisciplinary understanding of medicinal chemistry, computational drug discovery, and AI, driving advancements across the pharmaceutical field. Lately, Dr. Kumar has also served as an editorial board member of scientific reports in Springer Nature and Flanders in Molecular Neuroscience. With this brief introduction, I would like to invite our today's speaker, Dr. Rajneesh Kumar sir, to start the session. Thank you.
Okay, thank you so much, Gomati madam, for the kind introduction. Am I audible? Yes.
Yes, sir. Good morning.
Okay, Gomati madam, can you hear me?
Sir.
So thank you so much, madam, for the kind introduction, and I would like to express my gratitude to Durai sir as well. FDP actually. May I request everyone else to mute themselves so that
It's audible, sir.
I think someone's mic is still on actually. So I see, I hear, I don't know. Shivani Kavne, Shivani Kavne.
Arun NT and Shivani Kavane, please mute your mics.
Madam, as a host, I think you can mute. That will be.
आणि पेपर पण चांगल्या जर्नलमध्ये असंच करणार. रोज जाऊन जातेस ना कॉलेजची बसला.
Shivani Kavane, Shivani, can you hear us? Hey, Shivani, are you there? I think the host, Gomati madam, I think it is better that if you can mute all others actually, so that will be better actually, because I can see that
Suddenly,
I can,
automatically, somewhat the host is changed, sir. I don't know how to change. I think maybe Sarika, madam. Hello, Sarika, madam. I think she is in a host, and that's why, sir,
okay,
somewhat.
anyways, now I think there is no background noise. So let us
Yes,
continue
sir.
because we are running out of time actually.
Yes, sir. Yes, sir.
Okay,
Okay.
so thank you so much madam, for the kind introduction, and big thanks to Durai sir as well, the kind invitation. I'm sure that you have already heard a lot about AI and ML and how AI is changing the drug discovery pipeline and all. So today, the focus will be, my talk's focus will be on, generative AI, how we can use generative AI and what are those tools which can be used. What is generative AI? How we can use it is really disturbing actually. So I don't know. I cannot. Madam, can you identify who is host actually?
Now, I think Durai, sir. Please, Durai, sir, please.
Okay, good. Yeah, that's fine now. Now I think Durai sir has muted all of us actually. So okay, good. Thank you so much, Durai sir. So what we will talk about is we'll talk about quickly I go through. So this is my first slide I always keep because I really appreciate the Indian scientists for their motivation and their hard work. And Sir C.V. Raman in 1930, he got Nobel Prize in Physics for his discovery of the Raman effect. And this picture is when he received Nobel Prize in Stockholm, Sweden. Okay. And this is a place which is situated in Stockholm, Sweden, in Karolinska Institute, and called as Nobel Forum. And in this small building, there are several meeting rooms and a small auditorium where the committee members, they sit and decide to whom Nobel Prize shall be given in physiology and medicine. And when Alfred Nobel wrote his will, so he wrote in his will that all the money from his business shall be put into a foundation, and the annual earning shall be split into 5 parts and given to people who are working in the area of benefit of mankind, actually area of research for the benefit of mankind, like chemistry, physics, medicine, physiology, like that. And I've been fortunate to work at this institute called as Karolinska Institute, where this Nobel Forum is situated, and who decides to whom Nobel Prize shall be given in physiology and medicine for last 100 years. Okay, so I did my PhD back in 2014 from Punjab University, Chandigarh, and then was in at Stockholm, Sweden for almost 6 years. Have been to Japan for around like a couple of months as a GSPS summer fellow. And then since 2019, I'm working at Department of Pharmaceutical Engineering, IIT BHU Varanasi, where the pharmacy education started first in not only in India but in Southeast Asia actually. And that's a privileged institution as well. So this, I believe you already know what is a drug. So drug can be used for prevention, for cure, or for treating the symptoms. For when we are using it for prevention, we call it prophylactic treatment. When we are using it for curing, we call it therapeutic treatment. And when we use it for, treating the symptoms, we call it, symptomatic treatment. And unfortunately, 90% of, or 80, at least 80% of the diseases, they have just the symptomatic treatment. There is no cure to those diseases like Alzheimer's, Parkinson's, ALS, cancer, diabetes, right? So we are just healing the symptoms actually. And this is how drug discovery paradigm look like. So you start with a target identification, and then you, once you identify the target, uh, you need to, validate it. It should be validated. You should have a robust assay system. The structural information should be available if you are using structure-based drug design. And the lead inhibitors, are they available? If they are, then you can use, ligand-based drug design as well. Once we identify a target, the next thing is to identify the hits or the inhibitors. And here comes, the role of high throughput screening, which is classically used, that you screen hundreds of thousands of compounds to identify one which inhibit your target and which binds to your target. So in computational drug design, you can use virtual screening instead of high throughput screening. You use virtual screening where you use, computational tool to identify those hit compounds. And then there is another thing which is called as generative AI. So then you use generative AI so virtual screening is when you use the existing compounds, you identify inhibitors from the existing compounds. But when you are doing the generative AI, you are generating new compounds actually from scratch. Okay, so that is what, Gen AI can do. Once we get an inhibitor, it needs to be validated, and then iteratively doing all the chemistry, it get converted into the drug candidate, where the whole discovery phase is like really complicated and taking a lot of time. Success rate is very less, and a lot of failures are there. Similarly, once the drug passes through the discovery phase, it goes into the development where it becomes a drug candidate, and then it has to, go through the preclinical safety studies, GLP safety studies like mutagenicity, detailed pharmacology, acute toxicity, pharmacokinetic properties, subacute repeat dose toxicity or reproductive toxicity. And then it goes into the clinical trial where it is being tested in the humans, like in phase clinical trial 1, clinical phase 2, clinical trial phase 3, and finally it get registered. And the problem with this development phase is also that the success rate is very low, costing a lot of money, and taking a lot of time. So can we use AI to just squeeze in the time period instead of 10 to 12 years for discovering a drug? Can we reduce it to 2 to 3 years? Can we increase the chances of success of them? Can we decrease the failure rate? And can we reduce the cost? So these all three are the major objectives of using AI in drug discovery. And believe me, we can use AI at each and every step of drug discovery. Either it is target identification, it is hit identification, it is lead optimization, it is clinical trials, it is toxicity studies, ADMET properties prediction. So we can use it as each and every step. But since this lecture or this session is focused on Gen AI, so we will not talk about all those things, and we will focus on the Gen AI part in this one. And then before talking about Gen AI, I wanted to give a brief how to say a glimpse about how challenging it is to discover a drug. So this is a case study of discovery of this omeprazole. It's a blockbuster drug. Does anyone know what is a blockbuster drug? You can put a message in the chat box actually. Does anyone know what is a blockbuster drug? Okay, so a blockbuster drug is a drug which is making around 1 billion USD per year, and 1 billion USD per year is around 1000 CR. So if a drug is making 1000 CR every year, uh, that is called as a blockbuster drug. And then can um, imagine like how much money a drug can make, right? So the company AstraZeneca, they wanted to discover an anti-secretory compound which can inhibit the secretion of acid in the stomach. So they identified this compound called as CMN131, which was a pyridine derivative. But when they did studies on this, so they found that it was showing liver toxicity, so they could not use this molecule. So then they had to go to another molecule, which they identified was a benzimidazole pyridine derivative. But then they found that this compound was patented by some other companies, so they could not use that compound either. So then they have to find another compound, which they did in 1974, which was named as timoperazole, which was again a benzimidazole derivative. But the problem with this was that it was inhibiting the iodine uptake. So it was, toxic to the animals, and they could not proceed with it further. And then they came up with this molecule called picoperazole, which was again showing, severe toxicity in the form of necrotizing vasculitis. And finally, in 1979, they came up with this drug called as omeprazole, which was a potent and non-toxic and became a blockbuster drug. And then later, they found that this omeprazole contains enantiomeric sulfur, which can exist in S and R enantiomers. So then they found that, okay, S omeprazole, S enantiomer is more potent. So they started marketing that as omeprazole as a, new compound. Right. So now you can see in this whole story how difficult and challenging it is to discover a drug. So there are so many challenges, and then it takes, it took them starting from 1972. It took them, at least 7 years to discover this drug, when they had, all the, how to say, uh, facilities, all well funded, a lot of manpower, everything. So you can just imagine the difficulty of discovering a drug in academia, actually. Okay. So then I have this question for you. What is here? What will be there in place of the question mark? You can, as always, you can write your answer in the chat box.
10.
Yasmin, yeah, 10. Someone is saying 20. Okay. Yeah, I think everyone agrees that it will be 10, right? So I also say that it is 10. But can you answer this question? Which of the following molecules are water soluble? Or I can say, for example, which of the following molecules are, BBB permeable? So can you
Which
answer?
contain the carboxyl, which contain carboxylic group is a water soluble.
Okay, other than that,
So long chains, those are all insoluble.
don't tell me the rule actually that carboxylic acid containing group will be water soluble. Tell me which molecules are water soluble from this group actually, from these 5-7 molecules. Jasmine is saying that all look BBB permeable. Look means what actually? So can you say this with the same confidence you answered the previous question? For example, this question. Yes or no? No, right? So you can say that we cannot answer this with the same confidence by which we answer this question. And the reason behind this is that here we could identify the pattern. Here I can see that, okay, every time I'm adding 2 into the previous number, and then I can get this last number, which is 10. But in this case, I cannot identify that pattern until and unless I'm a seasoned chemist working in, solubility studies for like 30 years. I may find it difficult actually, right? But here computers can help us. Here, AI can help us. And the reason how AI can help us is because there are two things. One is that it is, very fast. It is very fast that it can process a large amount of data. Okay. And then second thing is it can identify the hidden patterns, right? It can identify the hidden patterns like which we cannot do. Okay. And then it works because computers, they work 24/7, so they are fast actually. So it is fast, and then it can identify the hidden patterns. So, and that is why you can get help from the AI. Okay. And this I'm sure this you have seen a lot in this 1 week. That AI is the big circle, and then we have a small part of it as machine learning, smaller one as deep learning, and then Gen AI comes in the end actually, which is a part of deep learning. Okay, so believe me, AI is in the air actually. So you have, for example, starting from the time when you wake up to the night when you go to sleep. So we are using AI in knowingly or unknowingly. Okay. Uh, Yeah, and then we coming to the challenge associated with drug discovery. So you see that there the chemical space is huge. So if you talk about the number of possible drug-like molecules, that is around 10 raised to the power 60. 10 raised to the power 60. Okay, and out of that chemical space, our objective is to identify one molecule bind to the Okay, so we have to, identify one molecule so that you can see like it is finding a needle in a haystack, right? So this is the biggest challenge actually, right? And then there are basically two kinds of approaches. One is called as direct approach of molecule design. So here on the top, you see this is a functional space where you plot, properties like you plot on this X, Y, Z coordinates. You plot molecular weight on X, you plot log P on Y, and then you plot solubility on the Z axis. And then this at the top of this hill, like Ooty is has a lot of hills. So like if you want to go to the hill, so that the top place of the hill is the desired functional space. Okay. So this is our functional space. And then we have this chemical space. For example, the existing 10 raised to the power 60 molecules, which are in the desired chemical space. Now in direct approach, what we do is like we synthesize one molecule, okay, or we, do DFT or calculations or single molecule. And then, for example, here we synthesize one molecule and determine its solubility using experiment. And then we plot it on this functional space and then we see where it lies. Okay. But there is very less chance that the first synthesized molecule will lie in the desired functional space, like which will lie on the top of the hill. Because it is just a random approach. We are synthesizing molecule and then we are testing them. So until unless we find a molecule which lies into this desired functional space, we have to, continue synthesis of those molecule, right? So this is called as direct approach. This is very random, very time taking. Another approach, molecular design approach, is the inverse approach. Here, we first, do some prediction. For example, we use virtual screening, and then we, try to select those molecules which are uh, potentially lying in this functional space, because we haven't done the experiment yet. We are just predicted it using either a QSAR model or molecular docking or any sort of similarity search virtual screening method, and then we identified that okay, this molecule can lie in this desired functional space, and then we do the experiment and then we test the molecule and then we get the molecule that okay, this is the molecule which is lying in the desired functional space. So this is called as inverse approach. Means first we are predicting the properties and then we are identifying, making the molecule. Here in the direct approach, we are first making the molecule and determining the properties. And then another inverse approach is using these, optimization methods or evolutionary strategies, or generative AI models like variational autoencoders, general adversarial network, and then reinforcement learning. So here what we are doing is we start with one molecule. For example, we randomly started any molecule. It is like reaching to the summit of a hill in a hill station. So you can start from anywhere. If you want to go to the top, you can start. There can be multiple starting points. So you start from anywhere, and then you try to optimize this molecule in this property space so that you finally reaches the desired functional space in minimal steps, right? That is the desired one actually, in minimal steps, because otherwise if there are 1000 molecule in this space, so out of those 1000, of course, some of them will be lying here in this functional space. And if you test all of them, you will find them. But that is not ideal, because you want to test minimal number of molecules to reach to that, desired functional space. Okay. How many of you are using ChatGPT or Gemini or Claude? I think most of you, right? So if I ask ChatGPT, can you suggest me some journals in the field of AI and drug discovery? It will give me a very nice list that, okay, these are the journals where you can publish something related to AI and drug discovery. But if I ask ChatGPT, can you generate structures of new drug-like molecules that are permeable to blood-brain barrier? So instead of generating molecules, it starts giving me, the parameters that, okay, size of the molecule is matter, lipophilicity matter and all, but it cannot produce, it cannot generate molecules because it has not been trained on that kind of data. Okay. But fortunately, there are tools, there are methods which exist, which can generate, molecules using generative AI. So what is generative AI? Basically, the ChatGPT has been trained on text. So it has read all the books, English, German, French, Hindi, Tamil, Telugu, right? Kannada. So it has read all the books and now it has learned all the language and grammar, and then it can build the sentences, right? So likewise, we can generate models which can learn the grammar of, molecules, learn the chemistry of molecules, and can come up with the new molecules. So this is called as de novo drug design, where we create new molecules from scratch, and then we use all these models. Okay, and then we can generate optimized molecules. Means we can generate molecules which have desired properties. Okay, and if you look at the advantage of using generative AI, so the biggest advantage of generative AI is the coverage. For example, when we do virtual screening, so we might screen a library containing 1 million molecules or 10 billion molecule or hardly 1 billion molecules, right? But this de novo design space is huge, because in virtual screening we are already screening existing molecules, but in de novo we are expanding the generation to unexplored, uncharted area actually. So it can generate completely new structure, completely new scaffold, which even does not exist as well, right? And that comes with a challenge as well, because if you generate molecules which are completely new, then the synthesis of them can be quite challenging. So if you go by ease of synthesis, then virtual screening wins. And if you go by the coverage, broad coverage, then the de novo drug design tools, generative AI tools, they wins this case. So what we can do with Gen AI, we can do the de novo molecule design. We can generate new molecules from scratch. We can do the lead optimization, taking a molecule which is already existing. We can modify it and optimize it, fine tune the structure. We can do the drug repurposing. We can find a new use for the existing drugs, and then of course we can go for the personalized medicine as well, where we can personalize, we can identify personalized drugs for patients based on their multi-omics profile, then their genetic profile. So then there are basically these generative AI drug design models can be classified into two. One is called as chemical language model. So they are like, they treat molecules like a language, like, you use the SMILE string, simplified molecular input line entry system. And then they also uses the NLP, natural language processing capability. And these are some of the examples, like, the SMILES VAE, reinvent, Camberta, and SMILES RNN, LSTM, etc. Sorry. And then there are graph-based methods, models. So these treats molecules as graphs. So where you have the atoms are considered as nodes, and then bonds, they are considered as is. And then they use this graph neural network for making the molecules. So there are like MolGAN, Graph VAE, Graph AF, JTVA. These all are various models which uses a graph for making graph neural network for making the new molecules. Okay. And then these are some of these models: variational autoencoders, generative adversarial network, reinforcement learning, recurrent neural network, transformer-based models, and diffusion-based models. I'll not go into detail of these. And then in the graph, in the chemical language model, as I said, like what happens is like you have this pyridine, okay, and then this pyridine is represented as miles in, you know, N1, CC, CC, C1, okay, and then you make a, you know, generative process where you each time you add one character and then keep on adding the next one to make this molecule, right? And then in the graph based one, so you have this molecule pyridine, same pyridine, and then here you represent it in the form of the nodes and then edge. So these are the nodes, these atoms are the nodes, and then these bonds are the edges. So and then you make, you know, starts from, you know, one node and then the next node and then add an edge between these nodes, and likewise you make the molecule and then you terminate the this prediction. Okay. Okay, well, yeah, and then there is another thing which is called as, you know, um, if you want to include the structure aware models, for example, the normal file based models, chemical language based models, they do not take into account the protein structure, right? So we can also incorporate the protein structure information. So from protein structure information, we can get the embeddings about these residues, and then you have the chemical library where you have the molecules, and then you can generate a molecule which is specific for that, you know, specific pocket. So in this case, this is called as structure based generative AI models, which can generate molecules which are suitable for that specific 3D structure, and that is also, you know, becoming catching up and then becoming popular. Okay, and then, you know, as I said, like the challenge with this deep learning, sorry, the generative AI models are that the synthetic accessibility is really a problem. So it can generate really new molecules which can be really difficult to synthesize in the lab. So that is why we use some, you know, the benchmarking metrics. We use some criteria like synthesizability or synthetic accessibility. So we do some scoring on those molecules whenever they are generated, and then we see that okay, these molecules is having a synthetic synthesizability score of, you know, out of 1 it is just 0.2, so it means that it will be difficult to synthesize. Okay, so these are some of the challenges of the in the generative models, like when we the limitation is with the evaluation metrics as well, like the synthetic feasibility gap, as I told you. And then there are, you know, over reliance on the proxy scores, because some of the times these are the simplified properties like quantitative estimate of drug likeness or log p. They do not reflect the real world drug discovery complexity. And then there are some, you know, problems in the distribution metrics as well, like FCD or KL divergence. They may favor chemically unrealistic distribution that mimic the training data superficially. And then there are problem challenges with the model overfitting and data bias, where the training set dependency is a lot. Like when you train a generative AI model on some specific data set, so the molecules it can generate will be, you know, similar to that input data set only. So that is called as copy problem as well, because, you know, some models they exploit trivial modifications. So you have, like, you give one molecule and then they just make small changes in that molecule and come up with a new molecule, but that is, you know, not innovative actually. Right, and then the real world relevance means, for example, you can design generative AI by using generative AI, you can design molecules, but when you go for synthesis, you find it difficult, and then you cannot, you know, experimentally prove it that it works. Okay, and then there are these algorithmic trade-offs as well, and then there are some benchmark design flaws as well, like you know, the lack of standardization and trivial task saturation. So now I give you some examples of, you know, these models. So for example, one of them is drug assist, which is a large language model for molecule optimization. So what it does is, for example, it works like ChatGPT only. So it leverages LLM for molecular design, and it also integrates chemical knowledge, chemical with natural language processing, and it enables intuitive interaction between the research and AI. For example, you tell it that okay, I have this molecule. Can you, you know, can you help me in adding one hydrogen bond donor to the molecule? So then it comes up with this solution, okay, that it adds one hydrogen bond donor in form of the carboxylic group, okay. And then you tell it that, okay, I want the log P value between 3 to 4, okay. So then it keeps that hydrogen bond donor so that it changes this carboxylic into this group, so that the log P is between 3 to 4, and then you can see that it says that, okay, now the log P is 3.28. So it says, can you generate two more molecules that meet the same requirement? And then it gives you two more suggestions that, okay, here also it has made some change, and here also it made some change. So it is just like a chat GPT where you can design molecule, you can modify molecule by using generative AI, which is LLM based generative AI. Okay. Yeah, so this is an example of the same tool where the quantitative estimate of drug likeness, means how close this molecule is to drug. It was 0.56, but then it generated a wrong answer, but then you use a human feedback, means you say that, okay, this is you says that, okay, you provided molecule is not correct, we found a molecule like this which is correct and similar to the molecule you provided, give me a molecule satisfying the requirement. And then it can improve that. So that is called as human in loop. So you keep human in loop, human feedback, giving back to the method, and then it works well. So this is an example of AI assisted discovery of anti-IBD lead targeting CXCR4. So in this case, they use like molecular docking. They identify top 200 fragment molecules, and then they use this initial fragment set to generate new molecules. And then using Gen AI, they generated new molecules. And then these were the predicted properties. And finally, they came up with some molecules which were really good targeting this CXCR4 receptor. Okay, then another tool is this is called as Drug Hive. So this is a structure-based generative AI tool where you can use the three-dimensional structure of protein. So you can use the three-dimensional structure of the proteins, and then you can generate the molecule. Okay, so this is how it works. So you have the data set which contains the experimental and predicted structures of the proteins and the drug molecules. Okay, and then you convert this ligand structure into the the features which are then encoded into this ligand encoder, and then you have the protein pocket information, and that you extract features from this protein pocket, and then you put them into the protein encoder, and then this and the model, it generates molecules. It generates molecules which fits well into this protein pocket, right? And that is what it is called as this structure-based generative AI design. And it has several possibilities. For example, you can do the substructure modification. For example, you have one molecule, and then you ask it to generate a molecule analogs by changing this part of the molecule. Okay, so it can generate the molecules by changing any of the part of that part. That's called a substructure modification. Then you have the linker design, where you can use it to design a linker to link two molecules, like you have two fragments which needs to be linked. So you can use these two fragments and link these two fragments by using the linker design tool. And then you can do the fragment growing as well, like you just have one fragment and you want to grow it, make it grow into the pocket. So that also can be done by using this tool. And then it can do the pattern replacement as well, where you have the core scaffold can be replaced by using generative AI. So in every module, what it does is in every task it uses generative AI to replace that part of that molecule and generate novel molecules. And then drug flow, it is another tool which is basically using diffusion modeling. So where you can perform molecular docking as well, and then admit prediction also it can be done. You can do the molecule generation using all these different models, like you can use with Castle linker, you can do de novo, and then you can do R group optimization. You can do the sidechain optimization. You can generate the linker, and then you can do the scaffold hopping as well. You can change the scaffold of that molecule. And then, basically it is for generative AI and as well as for predictive modeling. And then there is this graph invent. So this is a graph-based model which is developed by AstraZeneca again. So in this case, what you do is you convert those molecules into graphs first, and then it uses a message parsing network, MPPN, and then it can generate the new graphs or new molecules by using the structure of the input graphs. Okay, and then the most popular one is the reinvent, which I like a lot as well, and then we have been using it in our lab as well. So what reinvent can do is, for example, it has a generator model, and it has different generator models, like it can generate atom by atom, it can generate a library, it can generate the scaffold, it can generate the linker, link the two fragments, and then it can do the mole to mole as well, mole to mole generation, which is called as transformer based molecular optimization. And then once it generates the molecule, you can use the reinforcement learning. So once the generator generates molecules, that will be sampled actually, and then the sampled molecules can be scored. Now, can any one of you tell me what is reinforcement learning? Can any one of you tell me what is reinforcement learning?
It's a machine learning tool. It's a part of artificial intelligence, subset of artificial intelligence, and which is used in car driving and robots and games.
Yeah, that's very good, madam. But how does it work? Can you explain that?
Reinforcement learning, just one move forward and one backward. So with that, it will reinforcement learning. The same application is used for the car driving and navigation. So there it is helpful, sir.
No, but
Gaming and
those are application, but how does it work?
Trial and error basis, sir.
yeah, trial and error basis.
Sorry?
Trial and error basis.
Trial and error basis.
You mean reward and
Reward and
penalty.
penalties,
Penalty.
reward and punishment. Okay.
Punishment.
Yes, sir.
Yeah, so I give a simple example. For example, so machine learning can be divided into multiple types, right? It can be supervised learning, unsupervised learning, reinforcement learning, and then transfer learning, curriculum learning. So there are so many different types. Okay. So with any simple analogy. So for example, I have a daughter, and then we go to park, and she's just 2 years old, and she has never seen a cat or a dog. And first time she see a cat, and then she's curious, what is it? And then I tell her that, okay, this is cat, right? Next day, we see a dog, and then she asks, what is it? And then I tell her, this is a dog. So now this is called as supervised learning. So now the my daughter is now the agent here, the model, which is learning with help of the master. Means I am making her learn, okay, about the data. Next day, when we go to park and then she see a cat, so then she will recognize it that this is a cat. Okay, so this is how supervised learning works. So this is called as supervised learning. On the other hand, we have unsupervised learning, where I'm not helping her to learn. For example, if she see 10 cats and 10 dogs in a park, so then based on the features which she observed, she can put them into two groups, right? She can separate cat, she can separate dogs based on the features she learned, but there I'm not helping her, right? So this is called as unsupervised learning. And unsupervised learning is mainly for classification, means what is called as clustering. So you have huge amount of data, you just cluster them into one group or several groups. That is unsupervised learning. Like we use principal component analysis or TSNA, all these things. And then comes the reinforcement learning. Okay, so in reinforcement learning, what we do is now on first, it is a type of supervised learning. So first I told her about cat, second I told her about dog. Next, now on third day, if we see a cat and then she says it is a cat, so then what I will do is I will give her a reward. I will tell her that okay, tomorrow we will have the double park time, so we'll have 2 hours for play time actually. So that is your reward because you predicted it correctly. Okay, you identified the data correctly. On the next day, if she see a dog and then she sees she says that it is a cat. Now her prediction is, her identification is wrong. So then I tell her that okay, now you will get punishment because you predicted wrongly, and then so tomorrow there will be no play time. So tomorrow will not come to the park to play. So you can see now with this help of reward and punishment, what we do is we bias or we force our model towards correct prediction. Okay, so this is how the reinforcement learning works. So once the generator model generates the molecules, here the sampling is done. Now these sampled molecules are scored. Okay, and for scoring you can use multiple things, like you can use RDKit property prediction for scoring, like you can predict the log P of this molecule, molecular weight of this molecule, or you can use a QSAR model to predict the properties like solubility, permeability, toxicity. You can use the molecular docking, you can use pharmacophore, you can use ChemProp as well for property prediction. So now in all these cases, for example, I used the QSAR model and this generator model generated a molecule. I predicted its solubility using the QSAR model, and now the QSAR model says that the solubility is low. Okay, so then I give a punishment to the model. I will tell the model that okay, don't generate molecules like that because this is not soluble, right? And then if the solubility is good, the model will get a reward that okay, the solubility is good, you are doing well, keep on generating such kind of molecule. So this is how this reinvent 4 works, and this is a wonderful tool. I suggest all of you to try this actually. Okay. Yeah, and then there is dog stream that also comes from the what is called as AstraZeneca. So in this case, what it is doing is it is using reinvent. Okay, so it uses reinvent to generate molecules, and then the generated molecules, they are being docked by using dog stream. So they are being docked, and then this docking score is used as a score actually for reward and punishment in the reinforcement learning. So for that, they have already made a tool called as dock stream. And then in this case, you can use any docking tool, for example, Autodoc Vina, Schrodinger Glide, OpenEye, RDoc, CCDC Gold. All these can be used to generate the the molecule and use docking as a scoring function. Okay, and then DrugX is another tool, which is for again for de novo design using deep generative models, and it works as a multi-objective reinforcement learning framework. And it is developed by the Leiden University group and available freely on the GitHub. Well, okay, and then Gentral is another model, which is another very popular model, which is being used by the In Silico Medicine. This is a big company, very popular, very famous in AI-based drug design. So Gentral is generative tensorial reinforcement learning. So what it does is it uses self-organizing maps as reward functions, and then it generate molecules. And then so for example, this is the training where you use the chemical space, put it into encoder, you get a latent space, and then the generator is going to generate the new molecules, right? So in this case, it can use different self-organizing maps where these are being used as reward functions, and then they uh, define which molecules to keep and which not to keep. Okay. And then this is an example where the Gentral model was used to design molecules for DDR1 kinase. It's a kinase target. And then so they used, the, how to say, the database for selecting these reference compounds. And then they did a pharmacophore hypothesis, used pharmacophore model for as a scoring function. And then they prioritize these generated molecules based on all these properties, and they generated 30,000 structures. Out of those 30,000 structures, they selected 40 structures, and then they synthesized them, and then they came up with a nanomolar hit compound for this target. So you can see that, like, and this all, you can see that this from day 1 to day 46. In 46 days, they were able to, generate a nanomolar inhibitor, and that is patentable and potent actually. So you can see like how much time AI can save. Okay. Okay, coming to the summary, because I think we are on time actually. So the generative AI is revolutionized drug discovery by accelerating de novo drug design, optimizing molecular properties, and significantly reducing development timelines. And this AI has shifted the focus from molecule discovery to creation of entirely new compounds. And again, it is inherently interdisciplinary, requiring knowledge in machine learning, chemistry, bioinformatics, and computational biology. And I would like to highlight that this course, I don't know if some of you have already seen it on NPTEL. So this course I floated last year, and this year also it is being floated, and right now around 10,600 people have registered for it. And I think if you find it interesting, still I think there is some time to get registered. I think around 3rd or 4th of August is the deadline. So you can, request your students or whoever you want to register for this. So in this course, I have covered from target identification to clinical trial, all the AI tools I have covered. And then it is a 12 weeks course containing 60 lectures actually. Okay, and then with those words, I would like to express my gratitude again to Durai sir and Gomati madam for invitation and kind hospitality, virtual hospitality, I would say. Okay, thank you so much. If you have any questions, I am happy to answer or have discussion. Thank you.
Thank you, sir. Thank you, sir, for an enriching and intellectually stimulating experience. You have taken through the difficulties in drug discovery. You have taken us through difficulties in drug discovery till like how to accommodate all the difficulties and all. Now, I would request all the participants to ask any questions, any queries regarding the session.
Sir, good morning.
I think there is no queries, I believe, or
Good afternoon, sir. This is Dr. Sirisha from Vagdevi Colleges. It's really good to hear from you regarding this topic. It's really well. Thank you for a very lucid presentation. You made us, you explained the ML so well that we are really happy. Even the beginners are able to understand with live examples, real-time examples. It's a wonderful explanation, sir. Thank you to listen from you.
thank you so much.
Can you please repeat the difference between CLMS and graph-based models, sir? There I couldn't get it.
Yeah, so basically what happens is, as I said, like chemical language models, means chemical language model is basically kind of a language. For example, in English, we build, the sentences by using alphabets and words. Okay. So in the same way, we represent molecules by using smiles. So smiles is a representation where we use, alphabets and letters to represent alphabets and numbers to represent molecules. Like if I want to represent benzene ring, so I say C1, C, 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 C1. So that C is carbon and 1 is like it tells about the connectivity, right? So the chemical language based models, they work on the basis of language grammar actually, like what should come first in a grammar. You have like verb, adverb, adjective, noun, pronoun, all these things, and there is certain rule in every language that okay, this should come first, this should come at this place, like that. So likewise, chemistry be chemistry as a grammar, and that is represented in the form of smiles. So there we can use it, and then that is that capability these chemical language models has, that they treat it as a language. And then there are other tools which is called as graph based. So where instead of representing a molecule into a string of alphabets and numbers, we represent it as a graph. Okay. And then where we take the atom as a node, and then bond atom as a node and bond as an edge. So it's just like we make a spider web, right? So likewise we represent the molecules. And then so it is basically an advanced version of graph theory actually. So where you use graph theory to learn how the graphs are built, and then generate new molecules using the graph theory, using the graphs which were used as an input file, as compared to the chemical language model where it is using the language, like how, for example, carbon should have a valency of 4, okay, nitrogen should have valency of 3. So all these grammar rules are there in the chemical language model.
Okay. Right, sir. Thank you.
You're welcome, madam.
Sir,
Yes, sir. Basaraj, sir. Yes.
yeah, nice to listen your talk, sir.
Thank you so much,
Sir,
sir.
actually, for synthesis language is different. Talking and all in silico, whatever we are doing, that is different. Means, see, we are familiar to some reactions.
Yeah.
When we are doing some derivatives of basic molecules and drugs, no, we applied with some chemicals commonly. We can say as just one increasing on ester and making hydrazide and skip base. So with this only we are moving up. But in docking and all, it is giving a information how much pharmacophore should be there and what should be the length and all. So while doing all these things, we'll forget where we can't reach the chemistry to fulfill that. And synthesis wise,
Yeah.
technically we can say ingredients, all these are required. But when we are doing practically, we face so many problems.
Yeah.
Docking score will be different, and well, synthetically, if you have planned, that strategy is not working. So many
Yeah.
things. So, in that view, what you are going to highlight, sir, please?
Yeah. So thank you so much, Basavaraja sir, for raising a very important issue, right? So this is really important issue. The thing is that there are two things actually. The first one is the data quality. For example, I don't know if you have ever noticed that our weather forecasting app in India are not that much accurate. For example, we see that it will.
Hello, ಇವ್ರನ್ನ ಕರ್ಕೊಂಡು ನೀನು ಒಬ್ಬನ್ನ
So, for example, the weather forecasting apps are not very, reliable. So we see that our phone is showing that it is raining outside, but it is not raining. Okay. But if you go to other countries, for example, so there it is very accurate. So it says at 5:10 it will rain, so it will rain at 5:10. So that depends on the data quality actually. First thing is the data quality. If we have a very good data in our hand, and then the data we can use to make good quality model, those models will work very well. The another thing is when you say that okay, we can design any molecule, but synthesis wise that is a problem, we cannot synthesize, or the chemistry is very tough actually. So for that, people have been trying to develop new model, because to be honest, this generative AI, I can take a full 1 week course actually. So the other problem is that how to design molecules which are synthesizable. So recently a new tool came, Praxis in, which what it does is instead of generating any molecule, it will generate molecules only from the starting scaffolds you provide it. So you give it, for example, 10,000 amines and 10,000 carboxylic acids. So now these 10,000 amines and 10,000 carboxylic acid can make millions of amides. Okay. Now this generative AI model will generate only those amides which are suitable to your binding pocket, and then which you can synthesize as well, because it's just a simple amidation reaction, right? So I believe there are ways, but the most important thing is in computational chemistry and AI, the basic concept is garbage in, garbage out. Means if our input data is not good, our outcome will definitely be not of good quality.
Then how to validate, sir? After the model is generated, how to validate it?
Madam,
And
the only way to validate is to do wet lab.
that it is
So you generate models, you identify, you generate molecules, and then you synthesize them, you test them, and that is the only way to validation. There is no escape from wet lab validation. Because computational tools, they work as a companion. They can reduce, as I showed you the in silico medicine example, that in 46 days, 43 days, they were able to synthesize nanomolar inhibitor, but they designed, they synthesized, and then they test it, right? So then only you can make sure that okay, it works.
okay, sir.
And then some of you asked that whether these tools are open access. So whatever tool I have covered in my presentation, all of them are open access, because I am a big fan of open access tools. Because I don't want that. So I always give this example that open access is something like, if you are a good cook, you go buy or do your grocery, make nice meal and enjoy. On the other hand, you go to a restaurant and order food and eat it there. So the paid or the commercial softwares are like, you go to restaurant and you eat there, and open access is you cook your own food. So if you are a good cook, then of course you can make much better dishes than what you get in a restaurant, isn't it?
Yes, sir. Yes.
So then you have to learn cooking. Means you have to learn how to do all these things. So you have to learn how to use ML, how to use AI, how to do computational chemistry, and that takes some time, but it's not difficult actually. Anyone can do it. So all the tools which I have covered in even in NPTEL course are open access. You can access them anytime. It takes some time for you to learn them, but you can definitely use them and make good use of them.
Okay, sir.
Any other question? No?
Any questions?
Okay, then. So there is no question. I can express again my gratitude to Durai sir, Gomti madam, and Vasaraja sir for this, invitation and excellent event. I can see like 158 people have joined, so it's really great to talk to all of you. Thank you.
Thank you so much, sir, for your valuable presentation. Thank you, sir.
Do I need to stay in the call for the valedictory, madam, or?
Yes, sir. Yes, sir. Please stay, sir. Please
Okay,
stay.
okay, madam.
Now I request all the participants to turn on your cameras for a virtual group photo session. I request all the participants to turn on your cameras for a virtual group photo session. It is so Rajni sir, please turn on your cameras. Thank you, everyone.
Now, Steve.
Thank you, everyone. As we near the end of this insightful journey, we have arrived at the final milestone of our 5-day faculty development program on artificial intelligence in transforming drug discovery research, which was being conducted from 27 July 2026 to 31st July 2026, organized by the Center for Advanced Molecular Design and Simulation, Department of Pharmaceutical Chemistry, Jesus College of Pharmacy, UT. With the divine blessings of His Holiness Swamiji, I warmly invite the patron SP Dhanabal, Principal JSSCP UT, Convener Dr. R. Kalirajan, HOD, Department of Pharmaceutical Chemistry, co-conveners and coordinators, Department of Pharmaceutical Chemistry, JSSCP UT, and all the participants to stay with us for the valedictory session. Your active participation made this event a success. So please stay tuned as we celebrate our collective learning and wrap up this final chapter together. To formally bring this event to a close, it is my privilege to invite Dr. T. Durayanand Kumar Sir, Associate Professor, Department of Pharmaceutical Chemistry, JSSCPUT, to deliver the concluding remarks. Please, sir.
Is it audible now?
Yeah.
Yes, sir.
Okay. Sorry, again, I'm sorry. Hope I'm audible. I'm Dr. T. Durai Ananda Kumar. I'm here to give a concluding remarks of the online FDP program which we conducted for the last 5 days. And before going for the concluding remarks, I'm here to give an announcement from the Department of the Pharmaceutical Chemistry. We are going to organize a 2 days national seminar on recent advances in artificial intelligence for drug discovery and development from September 18th and also on September 19th. And it is a 2 days national seminar. In the same WhatsApp group, all the details will be shared. And I request everyone to be available in the WhatsApp group. Hereafter, we'll communicate all our department and college activities through the WhatsApp group. We can get benefited for that. The further details in detail will be shared in the WhatsApp group at the later point of time. And the 5 days FDP program was conducted in the banner of CAMDAS. It is our center here for the advanced molecular simulations with the support of JSSHR. It is all conceived by Dr. B. Suresh, Founder Chancellor, JSSHR. Currently, he is a Pro Chancellor of our university, JSSHR. And also Dr. S.P. Dhanapal, Principal JSS College of Pharmacy, Ooty. He supported all these activities, and we have established the Center for Advanced Molecular Design and Simulation. And we have designed to conduct so many activities, and one of the activity is to promote the faculty, research scholars, and their research experiences. In this backdrop, we would like to conduct seminars, workshops, training programs, consultancy service, all other related things. That is our prime objective, so that we also grow in research and we also support others to grow in research. So in that connection, the very first event of that is this EFDP program, for which we have identified 5 well-known, efficient, eminent speakers. The first day we had Dr. Prashant S. Karkar. He is a professor, Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai, Maharashtra. He has delivered a talk on foundational concepts of artificial intelligence in drug discovery. In fact, the talk was very elaborative. He covered almost all the aspects of the artificial intelligence and its connection towards the drug discovery research. And the second day, we had Dr. Chandan Shivamalla, Deputy Dean Research, Associate Professor, Department of Biotechnology and Bioinformatics, JSS Academy of Higher Education and Research, Mysuru, Karnataka. Along with Dr. Chandan Shivamalla, there were 4 people joined for the discussions. They all discussed on the topic artificial intelligence driven bioinformatics approaches for modern drug discovery, from target identification to drug repurposing. So it was more than a workshop. The Dr. Chandan Shivamalla team, they have taken everyone into the live. Followed by on 29th of this month, Wednesday, Dr. Perugu Shyam from the Department of Biotechnology, National Institute of Technology, Warangal, Telangana, delivered a talk on next generation AI-enabled pharmacophore modeling and 3D QSIR for computational drug discovery. And again, Sir made the sessions very live. The interactions from all the participants were really appreciable. And on the next day, it was yesterday, 30th of July, Dr. Chandrasekhar Ponnuswamy, Department of Biotechnology, Ratinam Global, deemed to be University, Coimbatore. He delivered a talk on artificial intelligence over drug design, from computational intelligence to next generation therapeutics. And Sir made it very elaborative, and it was for both for the beginners as well as for the researchers. And followed by today, we had a very eminent personality, Dr. Rajneesh Kumar. He is well known through NPTEL platform. He is conducting so many activities. In fact, Dr. Gomati Swaminathan introduced Dr. Rajneesh Kumar very well. And also Dr. Rajneesh Kumar introduced himself very well, and he don't require any introduction. In fact, he is from Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, Banaras Hindu University, Varanasi. He delivered a talk on AI in drug discovery, generative AI in molecular design and synthesis. In fact, no, we have requested all the speakers to deliver a talk on selective topics, which we have requested them. So in order to align the title of the workshop, the 5 days of EFDP come workshop, they all readily accepted for our request invitation. And all the 5 speakers made it very live. And they have given all the introduction to the online tools and how the research can happen. And some of them, they shared their personal research experiences that might have motivated every one of you to go towards the research and facilitated you to understand what is the basic element of artificial intelligence tools. And also, we greatly appreciate all the participants. Most of the participants, they were active in their sessions and they were responding to all the queries raised by the speakers of all the day, and it was very interactive. So that much only I have here. Before I conclude, I would like to play a video.
So it is not audible.
Okay, madam. See, sorry for that. But anyway, I can share this video in the WhatsApp group for the later time. And it's time to acknowledge all the support rendered by Dr. R. Kalirajan, HOD Department of Pharmaceutical Chemistry, Dr. B. Gaurama, and Dr. Gomati Swaminathan. They are the coordinators of the this departmental center. And also, I would like to express my thanks and record their support of Dr. S. Jubi, Dr. Gomati S., Dr. Srikanth Zupidi, our colleagues, for their support in organizing this event and making it very positive outcome. And due to the live coverage of Doordarshan in our campus, our principal and our HOD, they are not able to join. Otherwise, they were planning to join for this valedictory session.
Sir, excuse me, sir. Our principal sir and our HOD sir is there now with us.
Okay. Yes, I didn't notice. Sorry for that. They were busy with this thing. I thought that, okay. So with that, no, I'll thank once again all the participants for their lively participation. I'll hand over to Dr. Gomati Swaminathan. Thank you very much.
We both are available, Durai, for your valedictory.
Sorry, sir. Sorry, sir. I was thinking you are in the DD.
No, we were in PhD Viva. We came up. We came down for this program. Yeah.
Thank you, sir. Thank you.
Congratulations for the good program. I think good number of the participation across the country, and also good speakers you identified. Okay, for the last 5 days of the time, everyone had a very good time, and I could see the positive feedback, from all the participants in the chat box. So congratulations to you, and also Kali Rajan and all your team members from the Department of Pharmaceutical Chemistry. And I like to thank all the participants for making yourself convenient, attending the 5 days of this session. Thanks to you all. Looking forward to have you all on board in the future activities. Thank you so much.
Thank you, sir.
Thank you, sir. Thank you, sir. Your guidance and vision have been instrumental in making this program a grand success. All good things must come to an end. And as we conclude this enriching 5-day faculty development program, it is time to express our gratitude to everyone who made this journey possible. To formally propose the vote of thanks, I take immense pride in inviting Dr. Gomati Swaminathan to address the virtual floor. Thank you. Please, ma'am. Before going for vote of thanks, I would like to ask the participants to share your feedbacks regarding this 5-day FDP program.
Jasmine from Collegiate Pharmacy. This crucial time
Good morning.
2020 is about to start. We have to include in all of our teaching as well as research and all. I think to start with the research, it's a very good initiative in this crucial times. And this all the sessions were really informative. They got so many free tools information to start with. And we look forward for even collaborative research with many of the speakers who are on board. All the speakers were interactive and engaging during the session. It was very good information. The only fix was the mic muting and other things happening during the session. The host should have taken control over it from the beginning. That's the only small hitch during the session. Otherwise, everything is very good.
Thank you, Ms. Jasmine Abdulkar, for your effortless feedback. I would request any other participants to share your feedback on this 5-day FDP program.
Very good afternoon. I'm Dr. Anuradha from Sarojini Naidu Vanitha Pharmacy Mahavidyalaya, Telangana. Thank you, Dr. Danbal sir and Durayanand sir and all the team of JSS College of Pharmacy and all the speakers of this FDP. In this AI era, these particular sessions are very useful for the chemistry faculty, and this is very important to know about the artificial intelligence and all the programs in drug discovery, all the tools which are helpful. And today's session, especially the sir gave us all the tools which that we can learn, which are useful for all the chemistry faculty. So they can do the research and all the sessions were very, very informative and very insightful sessions. Thank you. Thank you very much.
So good afternoon.
Thank you,
one and
Dr.
all.
S. Anuradha ma'am, for sharing your feedback. Please, madam. Please, madam.
Good afternoon, one and all. I am Dr. Hemlata, Professor of Western Vanitha Pharmacy Mahavidyalaya, Hyderabad. So thank you, organizers, especially my special thanks to Durai Anand. So I think he worked a lot for conducting this FDP. And special thanks to all the excellent, eminent speakers. So they have given good information in AI. So based on that, we can develop a lot on new design drug molecules. So thank you one and all. So in future also conduct some workshops offline. Thank you, organizers. Thank you, Department of Pharmaceutical Chemistry faculty. Thank you.
Hello, good afternoon.
Good afternoon.
Madam, am I audible?
Yes, sir. Yes, sir. You're audible.
Yeah, myself, Dr. Vijay Kumar MMJ, working as a professor in the Department of Pharmaceutical Chemistry, SGM College of Pharmacy, Chitradurga. My special thanks and congratulations to Dr. Durai Anand Kumar Swami. Sir, Dr. Durai Anand Kumar, sir, who has been a friend to me since more than 10 years when I first met him during the time of the examination. And I was delighted to be the participant for this 5 days FDP program, which is very informative. I learned many things regarding the recent developments with the AI, and I wish all the very best. And I congratulate all the team members of this FDP program and to the college, to the management. And I wish many more such kind of FDPs to be conducted from this college. Thank you. Thank you, Randall.
Thank you, sir.
Good afternoon, sir. This is Sumita from Bageshwar Institute of Pharmaceutical Sciences.
I'm very thankful for organizers, especially Dr. Duday Anand sir. He was my teacher in BFOM. And sir, you have provided a good evidence. And the interaction sessions and with the faculty and the eminent faculty who are explained very well. And today's session was very useful, so informative. And from basics we have learned, sir. Very thankful, sir.
Good afternoon, all. Am I audible?
Yes, you are audible, ma'am.
Yeah, good afternoon, all. Myself, Kajal Saukule from Ashokramani College of Pharmacy, Petwadgaon from Maharashtra, Kolhapur. So, firstly, I congratulate all the organizers for this wonderful FDP. So, this FDP was so informative and well organized too. For the sessions, all the sessions are very engaging, very practical, which enhanced our knowledge. So I again sincerely thanks to all the organizers, all the resource persons for their valuable learning experience. Thank you all. Thank you.
Thank you, ma'am.
Sir, I have one question. Sir, if possible, can you conduct a hands-on training on AI, sir, for docking studies?
Actually, we are in the plan of conducting week-long programs in coming future. And slowly we will release the announcement on that. We are working on that. Week-long training program, not online, offline. Please be prepared for that. We'll all expect your presence here, having a fruitful research exposure. Thank you.
Sure, sir. Thank you.
Thank you, ma'am. Thank you, sir. Thank you, everyone, for sharing your feedback, as feedback is essential for growth, learning, and success. Now, we are moving to the vote of thanks. Okay.
Am I audible?
Yeah.
Thank you, sir. I make my humble pronoun to his lotus feet of holiness, and with his blessings, we commence the vote of thanks. Respected chief guest, principal sir, distinguished resource persons, esteemed faculty members, researchers, scholars, students, and dear participants. A very warm good afternoon to one and all. On behalf of JSS Academy of Higher Education and Research, Mysuru, and Department of Pharmaceutical Chemistry, and the Center of Advanced Molecular Design and Simulation, CAMDOS, JSS College of Pharmacy, UT, I extend my heartfelt vote of thanks to everyone who contributed to the successful completion of the 5-day online faculty development program on artificial intelligence in transforming drug discovery and research. I express my sincere gratitude to our distinguished resource persons, Dr. Prashant S. Kalkar, Dr. Chandan Shivamalla, Dr. Peruhu Shyam, Dr. Chandrasekhar Ponuswamy, Dr. Rajinish Kumar for their enlightening lectures, valuable insights, and generous sharing of knowledge. Next, I express my sincere gratitude to our respected principal, Dr. S.P. Dhanabal, for his constant encouragement, guidance, and unwavering support in making this program a grand success. Thank you, sir. My heartfelt thanks to our respected head of the department, Dr. R. Kalirajan, for his visionary leadership, motivation, and continuous support. Thank you, sir. And I also extend my sincere appreciation to all the faculty members of the Department of Pharmaceutical Chemistry, Dr. S. Jubi ma'am, Dr. Gaurama ma'am, Dr. Gomadi Sanish ma'am, Dr. Durayanand sir, and Dr. Srikant Chupadi for the dedicated efforts, teamwork, and commitment in organizing this event successfully. Thank you, sir. And I gratefully acknowledge the support of the management of JSS Academy of Higher Education and Research, Mysuru, the organizing committee, research scholars, and the technical support team, and all the staff members whose contributions ensured the smooth conduct of every session. Finally, I extend my heartfelt thanks to all the participants for your enthusiastic involvement, active interactions, and commitment to learning throughout these 5 days. Your participation has made this program truly meaningful and successful. Thank you, madams, and thank you, sirs. Once again, thank you all for your valuable support and cooperation. I wish everyone continued success in your academic, research, and professional endeavors. Thank you. Thank you, one and all.